It's my pleasure to introduce Professor Ariel Weissman. Professor Weissman graduated the Hadassah Hebrew University Medical School in 1988. In 94, he completed his residency in obstetrics and gynecology at the Kaplan Medical Center, Rehovot, where he spent another 2 years working as a senior physician at the IVF unit. Professor Weissman then pursued a 2-year research and clinical fellowship with Professor Bob Casper, the Division of Reproductive Sciences, Department of Obstetrics and Gynecology at the University of Toronto in Toronto, Canada. His main focus of research was transplantation of human ovarian tissue in immunodeficient mice. Upon returning to Israel in 1998, Professor Weissman joined the IVF unit at the Department of Obstetrics and Gynecology at the Wolfson Medical Center, where he currently holds a position of an associate professor. Together with Dr. David Gardner, Colin Howes, and Zeev Shoman, Professor Weissman has published the textbook of Assisted Techniques, Laboratory and Clinical Perspective, which is one of the leading books in the field of ART. And today, Dr. Weissman is going to talk about the results of the Virtual Academy of Genetics questionnaire on mosaicism and PGS.
Thank you for the kind introduction. Good afternoon, and thank you for the kind invitation also to the organizers. I would like to present the results of the Virtual Academy of Genetics questionnaire on mosaicism and PGS, which, as you will see, highlights very well the controversies on this topic that have been already raised. So IVF Worldwide is one of the largest internet-based platforms in our field. It contains, among others, a clinic locator, an education center, a newsletter, surveys, as this one, blogs, forums, the Genetics Virtual Academy, physician may contact and consult physician, and online journal, and others. And it's an excellent platform to conduct surveys because of the large number of clinics, of units that are registered from all over the world, all 5 continents. And it is also interesting because we receive data from both academic and non-academic practices on a global scale, which actually reflects the wisdom of the crowd. So again, it is not evidence-based medicine, but from these surveys, they are very frequently cited in this meeting as well, and in many papers. You get a very good feeling of what is going on in our field. And there have been many surveys that have been already published, conducted in the website. Many of them were published in peer-reviewed journals, which reflect the interest in those surveys. So, and the Virtual Academy of Genetics is one of the recent additions to the IVF Worldwide. It contains a lot of educational materials written by leaders in the field, and also publications such as position statements following the CoGEN meetings. This is the statement on PGS from the first CoGEN meetings 2 years ago, and the statement on mosaicism from last year's meeting here in Barcelona. So embryonic mosaic aneuploidy is a hot topic, and it has been the subject of many publications, many debates, which prompted us to conduct these surveys. These are the guys that composed the survey. You should know them by now. And the purpose was to evaluate the extent of mosaicism in PGS clinical practice and to gain an insight on the views and practices of the ART community regarding this issue. It was posted during the month of December last year for 1 month only. And the title was Survey on Mosaicism in Preimplantation Genetic Screening. What is your opinion? And we have techniques for quality assurance, to minimize duplicate reports from clinical units, and to minimize possible false data. Overall, it was the month of December, 1 month. We got responses from about 100 units, representing about more than 100,000 annual cycles from all over the world. The pointer is not really effective, but the majority from Europe, followed by North America, Asia, and also other parts of the world. And the majority of respondents, 92%, came from clinics that are performing PGS, and only 8% from clinics and clinicians that are not performing PGS, so there's a huge bias here. Questions were directed separately to those that are performing and those that are not performing. We're more interested in the views of those that are performing PGS. And the results, most of the results that I'll present are from clinics that are performing PGS. First question was, at what stage of development are the majority of embryos biopsied in your center? So 90% do the biopsies at the blastocyst stage, trophectoderm biopsies. Interestingly, a year earlier, we had another survey, a general survey on PGS, and at that time, the percentage of trophectoderm biopsies was only 72%. So you can see that within 1 year, there was a shift towards trophectoderm biopsies. Second question was, in trophectoderm biopsy, what is the optimal number of cells that you aim to obtain? And the majority, 60%, answered between 5 and 7 cells. In the CoGEN position statement on chromosomal mosaicism from the last meeting, the recommendation was actually the good practice recommendation that was that for reliable detection of biological mosaicism, a minimum of 5 cells, but importantly less than 10, should be biopsied with as little as cell damage as possible. And this is also the recommendation of the PGDIS. And there is not much published on that. There's this study by Neil et al., who took the DNA content. It is very difficult from following a trophectoderm biopsy to know exactly how many cells you have. So Neil et al. Took the DNA content as a surrogate for the number of cells that were biopsies, and they could show that trophectoderm biopsies with the highest relative DNA content were associated with a lower live birth rates after a single euploid embryo transfer. So this is support for being cautious with the number of cells that are being biopsied. Next question was, which method is predominantly used in your center for PGS? And we could see here that NGS was used in 60%, whereas RACGH in 30%. And a year earlier in the survey we conducted in 2015, end of 2015, next generation sequencing was only used in 60% of the clinics, whereas 60% were using RACGH. So there's also a big shift here towards NGS. And next question was, do you think next generation sequencing is required to reliably detect mosaicism? So here you see the results separately for clinics performing and not performing PGS. The darker blue are for those that are performing. So 60% said yes, and about a third of both said simply that they don't know. Well, NGS has brought embryonic mosaicism to the spotlight, and RACGH is considered now less suitable for establishing the exact level of low-grade mosaicism compared with CGS. Next question was, where is genetic testing being performed in the majority of cases in your center? So about a third use an in-house genetic laboratory, whereas 2/3 send their biopsies to a centralized referral laboratory. An interesting question, do you receive information on mosaicism from the laboratory? So in about a third, the answer was no. samples are reported as euploid or aneuploid only. So no information is given on mosaicism. Whereas in other cases, there are the mosaicism is reported, but with different degrees of details. There's no common or standard way of reporting, and this highlights the need for uniformity and standardization in reporting practices. So there's a big gap here. And if we try to differentiate between those who perform the biopsies in-house and in centralized laboratory, there could be perhaps a trend that in-house laboratories supply more details, but this is, I'm not sure how significant this is. Next question was, what is the percentage of mosaic embryos that you encounter in your practice? A third said, I don't know. And the majority, about 50%, said less than 10%, and others 12 or 8%, 11 to 20%, 12% answered, and 8% answered more than 20%. And this is interesting because we've heard also in this conference that there are differences in mosaicism levels between clinics, between units. So this may be a reflection of that. Do you ever re-biopsied mosaic embryos? 2/3 said no. And a third said yes, or under special clinical circumstances. At what level of mosaicism would you consider embryos as abnormal? Again, about a third said, I don't know. 26% of those who are performing PGS and 35% of those that are not performing PGS. The majority said more than 20%, and this is also in line of the recommendations of CoGEN and PGDIS. Should mosaic embryos be stored for potential therapeutic use or discarded? About 15% of those performing PGS said that they should be discarded, whereas the rest said yes, they should be stored for potential therapeutic use following individual counseling. And should mosaic embryos be transferred? 15% said no, mosaic embryos should never be transferred, where here there were a few possible questions, so the total I think is more than 100%, but under certain condition the majority said yes, they could be transferred. Yes, if no euploid embryos are available for transfer, yes, but only after mosaicism degree is below a certain threshold, and also depending on the specific chromosomes involved. So what kind of mosaic embryos would you consider for transferring? Again, some said I would never consider transferring mosaic embryos. And the majority, 60%, said it would depend on the specific chromosomes that are involved. And there are recommendations of both in the CoGEN position statement from last year for transferring mosaic embryos. I won't go into details, but from recent papers and what we've heard in the meeting, they should be perhaps reevaluated and refreshed, especially prioritizing the embryos for transfers. These recommendations are available, but perhaps should be refreshed and updated. How would you follow a pregnancy following transfer of a mosaic embryo? The majority recommend invasive prenatal testing. 35% recommend amniocentesis, and 18% recommend invasive testing by PGS. 15% like any other IVF PGS pregnancy. I don't know what that means. 24% recommend NIPT. and 8% recommend special genetic counseling before making such decisions. There are recommendations in the CoGEN position statement. If ongoing pregnancy is established after transfer of a mosaic embryo, the developing fetus should be tested for aneuploidy by prenatal diagnosis, and amniocentesis is recommended over CVS as the mosaicism persists in the trophoblast. And of course, reproductive outcomes of mosaic embryos should be collected and transferred with follow-up on the children. Which of the following statements best describe your views regarding mosaicism in preimplantation embryos? So there's a huge polarization here between those who perform and those who do not perform PGS. Those who do not perform PGS, perhaps this is one of the reasons why they are not performing PGS, state that they agree to the statement that mosaicism is a serious problem that puts in doubt the entire concept of PGS. Until the issue is resolved, the use of PGS should be avoided. Whereas the majority of those performing PGS agreed to the statement, joined the statement that mosaicism may be an issue, but has always existed in IVF. More study is needed, but PGS should continue with appropriate safeguards and counseling. Last question, I think. Which types of studies would be most valuable to better understand mosaicism? So the majority here also of several answers were possible to accept. So the majority would like to see more prospective studies about the impact of mosaicism on clinical outcome, as well as Darren has stated, distribution of mosaicism throughout embryos donated for research, multiple biopsies for each embryo, and between trophectoderm biopsy and the ICM. So to summarize, embryonic mosaic aneuploidy is common in preimplantation embryos. New techniques used for comprehensive chromosome screening, particularly NGS, can now detect its presence with high accuracy. More studies are needed before definite conclusion can be drawn regarding detection, clinical significance, and management of embryonic mosaicism in the context of PGS, and there's a definite need of, urgent need of standardization of reporting mosaicism in PGS by the laboratories that are doing the analysis. And these results generally support the CoGEN position statement on chromosomal mosaicism detected in preimplantation blastocysts, published after last year's meeting. If you are interested to read in more detail, this survey was recently published in the May issue of Fertility and Sterility. And this is a very nice, well, we're talking about mosaics. It's a very nice mosaic, recently recovered in a Roman villa, 1 mile away from my home in the old city of Lod in Israel. It's been already on display in the Metropolitan Museum in New York and in the Louvre, and you can find more details in this reference. Thank you for your attention.